I feel like that's not.
Tell you what, you want to go here and I'll. Are we live?
Mm-hmm.
Okay. Hello, school community. Thank you for joining us for our 2025 live stream presentations of our environmental science honors classes field research experience. They're going to be presenting on their findings where they explored a question at Rodeo Lagoon in the Marin Headlands. I'm going to pass it on to MK Schumacher, who is with Headwaters, to describe a little more about the project.
Hello, everyone, via stream. I had the pleasure of working with these students over the past 2 weeks. I came in last Monday and we did a question asking hypothesis development and methods development class. I brought in all of the research tools that we have available, introduced that to them, and then they were able to choose the topics of interest for their research project. Last Wednesday, we collected data in the field at Rodeo Lagoon, and then I came in class on Thursday and Friday, accompanied by my coworker Bria Gillespie, and we analyzed their data for their presentations, and then they are presenting today. I've had so much fun working with these students, and their presentations are amazing. So without further ado, we're going to get started with our first presentation. If you are watching the live stream and you have any questions, feel free to write them in the chat, and we will put them on the board at the end of the presentations. Thank you. All right, group one.
You guys start.
I'll go.
Okay.
Should I look at the computer or at the class?
Does not matter.
Okay.
As long as you're in the camera, you can look wherever you would like.
Okay. Hi, everybody. Our project is called Let's Bug Out, and we researched the effect of pH and salinity on macroinvertebrates. And with me, Ava, Summer, and Nikki. To start off with some background, the independent variables that we measured were the pH and salinity. The pH is a measure of how acidic or basic water is on a 0 to 14 scale, and the salinity is a measure of the concentrations of dissolved salts in water in percentage, which is parts per thousand. The dependent variable that we measured in our experiment was the score of the macroinvertebrate based off of the pollution chart, which Summer will explain.
So we were given an identification chart with 4 groups, 1 being very intolerant to pollution and 4 being very tolerant to pollution.
Okay, go to the
Oh,
camera.
I have to. Oh, sorry. So from there, we would categorize each type, and group 1 would be given a score of 4, group 2, 3, group 3, 2, and group 4, 1. So if you were to have a higher bug score, that would mean that your ecosystem is healthier, since bugs that are more intolerant to pollution can thrive.
Our hypothesis was as pH and salinity levels become more extreme, the diversity of species will decrease. And on the contrary, our null hypothesis is that there will be 0 correlation between the macroinvertebrates and the pH or salinity of the water.
Okay, so moving on to our materials and methods. So for our methodology, we chose to look at 5 different locations around the lagoon. One person took 200 strides between each location, trying to keep an equal distance from the shoreline. Salinity and pH were both measured using a water meter 2 times per each location, and then the macroinvertebrates were sampled 2 times per each location using a D-net and getting the sediment from the bottom. The macroinvertebrates were then sorted into a ice cube tray using spoons and droppers, and then we identified them using the bug chart. And the bug score was taken 2 times for each location.
It's this.
Okay, and then here's a visual representation of where all of our locations were.
Okay, now getting into our results. So our first graph depicts the relationship between the pH and the bug score, and we can see that the macroinvertebrate diversity peaks at around a pH of 8.1, and it also decreases at lower and higher pH levels, creating sort of this parabola shape. Our next graph is between bug score and salinity, and we can see that as the salinity levels rise, so does the macroinvertebrate diversity.
Okay. So wait, should I go? Okay. So moving on to our discussion, we first want to compare our results to our hypothesis. So we concluded that half of our data, which was the pH results, did support our original hypothesis, meaning as pH levels become more extreme, the diversity of species will decrease. So as we can see on our graph and what someone already explained, we do see that parabolic trend where we have very low diversity at the extreme levels of pH, which is the left and right hand side of the graph. And then we also see that higher diversity, which is the highest bug score or peak within our moderate pH range. Moving on, we also found that or concluded that our other half of our data, which was the salinity results, more supported our null hypothesis rather than our original hypothesis, meaning that there was no correlation between salinity levels and macroinvertebrate diversity. Again, we do see this upward trend rather than the parabolic trend that we originally were trying to see, where we have bug scores staying fairly consistent from moderate to higher ranges of salinity, with, again, that slightly lower salinity sites also having lower diversity.
Just to compare some of our results to kind of like the biological context of the field site, to reiterate, we were at Rodeo Lagoon, which is a very shallow brackish coastal lagoon, and it's separated from the Pacific Ocean by a sandbar. So this sandbar really made the pH and salinity fluctuate. Salinity is heavily influenced by when the ocean kind of overlaps with the lagoon, creating more salt water in it. And the pH is also influenced by different biological activities, such as the phytoplankton and the summer algae blooms.
Okay, so moving on, we did some of our own research on current science on pH, and we found that multiple studies citing extreme values of pH correlating to a lower diversity of macroinvertebrates. More specifically, we found a quote saying that altered pH affects macroinvertebrate survival by weakening shells and exoskeletons or reducing the survival of alkaline intolerant species. Again, we also found another quote saying that more of broad ranges of pH and their effect on macroinvertebrates, a study citing extreme values as below 5 or above 9 being more harmful, and the more moderate or natural range being between 6.5 to 8.5. And yeah, so moving on to another trend.
And these results aren't just applicable to macroinvertebrates. The picture right here shows that a higher diversity of any organism is achieved in the moderate or natural ranges, which are these three boxes in the middle. And whereas on the left and right, those are lower diversity, and those correlate with more extreme pH ranges.
So on to comparing what we saw in our data to our research. Similarities that we saw were that the higher bug scores happened in the natural or moderate range of pH, while our lower scores were found at the more extreme values on either side of the graph. So that kind of aligns with the idea that the bug diversity declines at more stressful levels. So for differences, the lagoon that we were at didn't really have extreme pHs. Anything that was above 6 or above 9 just didn't happen. And so that shows that our decline was more gradual rather than as sharp as we would see in more extreme conditions.
Okay, so now shifting to our second half of our data, in terms of research for current science on salinity, we found that there are two different types of tolerance for macroinvertebrates. They can either be stanohaline, meaning that they have a narrow salinity tolerance, or urohaline, meaning they have a wide salinity tolerance. On our graph to our right, we can see more of a species-specific response, and technically, yes, there is that increase or thriving of the macroinvertebrates within the moderate range, but at the same time, we can also see how more sensitive species decline, whereas on the other hand, we can have more salt tolerant or urohaline species increase as the more stressful levels increase as well.
And to kind of connect the results to current science, as we can see a similarity here from our data where there is a peak in diversity at a moderate level of salinity, and that also shows the same on the graph to the left.
Okay, and moving on to our differences, on the other hand, we did not see a big range between our salinity values. As we can see, our salinity values were between 7 to 8 parts per thousand, whereas the graph on our right is between 0 all the way up to 100. So again, that was one major difference, meaning that we did not capture extreme salinity events. Additionally, we did see more of an upward trend, and that could be depicted more of just on the lower left-hand side of that right graph. That's basically what we're seeing with that increase. And in the big picture, yes, there is an extreme or prolonged high salinity conditions may still reduce richness. However, our study reflects mostly moderate or tolerable ranges.
Okay, now going into some predictions. If we were to see an increase in sea levels, this would obviously increase the salt water in the lagoon, increasing the salinity, which would most likely decrease the diversity of macroinvertebrates since less can live in higher salinity levels. Similarly, for an increase in temperature, this could affect species that thrive in cooler temperatures, and it could also lower the pH due to a process called ocean acidification. So if the temp increase was caused by increased levels of CO2, the ocean would absorb the CO2 and turn it into a carbonic acid, which then would break into hydrogen ions, decreasing the pH. Moving on, if there was an increase in storms, this could increase the amount of fresh water, but it could also disrupt habitats due to the movement of sediment. So we would ultimately see a lower diversity of macroinvertebrates and a lower bug score.
As Summer alluded to earlier, the specific attributes of a lagoon ecosystem played a key role in the pH and the salinity results that we found. Most importantly, lagoons are enclosed water ecosystems, which makes them more vulnerable to human impacts like runoff and pollution. And their shallow water means that the temperature changes fast, and only a lower diversity of species are habitable to those changes. The high variability in salinity is also caused by the sandbar, like Summer was talking about, and other narrow inlets that are easily changed by storms and seasonally. And that impacts the salinity because the freshwater and seawater balance will change. As a land manager, there are measures you could take to continue keeping the pH in a good range. For example, you could implement year-round pH monitoring, which would make sure that you get a wider range of pH and salinity with the seasonal changes that we observe, because our data was very specific to how the ecosystem is in this moment. And also limiting the use of fertilizers and pesticides near the water source would prevent the algal blooms that Summer was talking about earlier, which would increase the soil acidity and the water by default. So that would eliminate that. And also removing obstructions from nearby freshwater streams would prevent the salinity fluctuations from decreasing.
Okay, so moving on to our caveats and future research. So some of the caveats that we had was our small sample size. So if we were to do a further study, we would definitely up our sample size from 2 to 4 and take more samples of the pH and of the macroinvertebrate score. We would also make sure to standardize the distance between each of our points because we use strides, and that kind of left room for human error. So if we were to do the study again, we would use a transect tape and also make sure that we kept an equal distance from the shore each time. We would also log the temperature and the cloud cover for each of our locations because as the day went on, the sun came out, and that could have also affected the macroinvertebrates that we saw. And we would also, because of the human error, some of the bugs might have been missed from each of the sediment scoops that we saw, and some of them didn't even show up on the bug score pollution chart. As we said before, the salinity levels of the lagoon change seasonally, so it would be helpful to add year-long samples. Okay, so given our low variability in our salinity data, we would also want to do a follow-up question on the effect of dissolved oxygen on macroinvertebrates, as our pH data strongly supported our hypothesis. For context, macroinvertebrates need oxygen for respiration, and low dissolved oxygen kills sensitive groups. Dissolved oxygen and pH are also indirectly linked because as photosynthesis happens, the amount of dissolved oxygen rises and reduces CO2 levels, which makes the water more basic or have a higher pH. This means that the pH and the bug score might be indirectly linked and caused by the dissolved oxygen, not the pH directly. So adding this would separate possible causation and correlation. So, yeah. Yeah, that's it. And then here are sources.
Does anybody have any questions about our project?
Will we also see it
Yeah, Lucas?
online?
So as you said, as salinity increases, biodiversity increases, right? So then does that continue on to a lot higher salinities, or does it eventually, do you think it'll level out?
So based on our research, sorry, I'm just gonna go back a few slides. Here, this was like a graph from like a study, and again, you can see that correlation. Yeah, yes, there was that increase, but basically in our results, we concluded that we were more of capturing just kind of that first half between the 0 and 10 for the Eurohaline species. So that's where we saw that linear increase. But in like a more broad idea, like technically, yes, there is that same like parabolic trend that we saw within the pH results. So again, it was just our narrow range that kind of like messed up, I guess, that whole idea. But yes, to answer your question, there is that.
Yeah, with
upward and
salinity
downward trend.
and pH, like either extreme isn't beneficial for species diversity.
Mhm.
Yeah, Annie?
Based on your research, would you say that salinity or pH has more of like a dramatic effect, or like which one should we be like more careful about to like protect biodiversity?
Well, as far as human impacts go, I think pH is more directly correlated to like the actions of humans around the lagoon ecosystem, but salinity is just because it's a variable and it's a unique ecosystem. Yeah.
Right. Yeah, it's like basically it depends. Like salinity more depends on like the contact within like the ocean. That's where it gets a lot of its like I guess like salt like amounts within the water to create either like high levels of salinity or not. So yeah. Mateo.
Was there like a trend of salinity like in terms of like the location in the lagoon? Like for example, was the area like closest to like the inlet higher salinity, or did it not really matter?
It was relatively like random, so it.
Okay. I think.
Okay, so you want to use up the time, or you have a quiz question?
A quiz question. I don't know. Do we have a quiz question? I can come up with one.
Or any other questions?
Yeah. You guys can ask like any like clarifications too if you want. If anything was like confusing.
What was your favorite part of your research?
That's that question.
I personally just like seeing like our data on the table. I know that's not research, but I just found it really interesting to kind of like see how the data we collected on Wednesday like did make a trend and how we were able to do that in just one day. So that was something I liked.
That was part of your research. That's a good
Okay.
All right.
Okay.
Thank you guys. Thank you very much.
Thank you so much.
Is this one yours?
No.
Yeah. That one? Yes. Okay.
Wait, it just follows my train of slides. Okay, yeah. Oh, yeah. Sorry. I don't get my big.
Just a reminder, if there are questions that they send in the chat, those will be displayed on the board. So you didn't have any questions on the board?
They'll appear on the
Yeah.
All right. Okay, so whenever you guys want to start, I'll listen.
Okay. Good. Yeah. We're good. Yeah.
You can start out.
Oh, okay. Good morning, everybody. Today we're going to walk you guys through the nitro bugs, so the effect of nitrogen compounds on microvertebrates. This is by Sabrine, Emma, Shane, and me, Andrea.
Wait, what? Oh, okay. So this is our group. So as Andrea said, we studied the effect of nitrogen compounds, which includes nitrite, nitrate, and ammonium on the presence or bug score of macroinvertebrates. So our independent variable was the nitrogen compound we were testing for with the little test strips in parts per million. And so this included NO2, NO3, and NH4. And then our dependent variable was the bug score, as the last group talked about. So our hypothesis was that both low and high levels of nitrogen compounds will decrease macroinvertebrate presence in the water and diversity of macroinvertebrates. And our null hypothesis was that nitrogen compound levels would have no effect on macroinvertebrate presence. Okay, so just for a little bit of background, because I feel like we've talked about this a lot, but this is an example of how the nitrogen cycle moves, particularly in like a water ecosystem. So it's moving through with this is like really confusing, but like look at with the ammonium and the ammonium right there, and then you have the nitrite where over there, and then the nitrate NO3 right there. And ammonium comes from the waste and dead plants of the animals, and bacteria change ammonium into nitrite and then nitrate. And plants and algae use nitrate to grow, and some bacteria turn nitrate back into nitrogen gas that returns to the air.
Okay, so you may be wondering how exactly we get this data. So the materials we used to collect this data were a D-net, some test strips, clear buckets, metal spoons, plastic droppers, ice cube molds, a bug tray chart, and then magnifying glasses, which we didn't end up using because they were big enough for us to identify. But this is just some in the field showing how we use this. Okay, so for our bug score chart, you guys already explained it, but this is group 1, group 2, group 4. Group 4 is the most tolerant to pollution, so that means they can thrive and do great in pollution. Unlike group 1, which is very we will not find any group 1 in very polluted waters. So when we scored them, we got 4, 3, 2, 1. So if you got a 1, that means you just found some species that were pretty tolerant to pollution. For a DNet, that is exactly how we scooped up our soil and macroinvertebrates. So you just kind of kick the soil a little bit, scoop, and then we put them in our clear buckets. And these were the test strips we used to test all 3 of our properties. So yeah. So these are our locations. We had 5 locations in the time that we were given. So these locations were about 85 meters apart. There were hundreds of Shane's strides. So that's how we measured it. Yeah, they were pretty equal in distance, but around 85 meters. And we know that because a different group was testing and they were ending in the exact same location when they actually measured it with meters. So for our methods, we first started by collecting NO2, NO3, and NH4. So we used our test strips, and NO2 and NO3 were used. We would dip them into a cup of water, which we would just take from the lagoon. We'd leave it in and swirl it for 2 or 3 seconds, and then we would lay it out on our clipboard for 30 seconds. We would wait for those 30 seconds to pass, and then we would compare it to our chart and see the levels. And then NH4 was a little bit longer, but we would do the same beginning, scoop a fresh thing of water, dip it in, swirl it for a few seconds, and then we would lay it out for 3 minutes to wait for that test to be completed. Our test strips were tested in ppm, so that was the units that we tested all of our data. And then we did these tests twice per location, so we had 2 of each test at each location to make sure that our results weren't just based on coincidence or were just a little bit flaky. So the second thing we did was we tested for macroinvertebrates. So this is an example of Andrea being in the water and scooping up our soil, water, and macroinvertebrates. We also did 2 tests per location. So we had 2 plastic buckets. We scooped in. Each bucket got 1 scoop. Then we would take the time, go through the buckets, collect all of our macroinvertebrates, put them in our ice cube trays. Then most of the scoop was sediment. We were able to use spoons and droppers to sort and get all the bugs. It did take much longer, but that time was very worth it because we were able to collect all of our macroinvertebrates. Then we would spend some time identifying them using the bug score chart that I showed and explained earlier. This is just us sorting. Again, we sorted, then we recorded the score. Each group, as I said earlier, has a score, and we would do it. Though many of the bugs and macroinvertebrates we found were not on the score sheet, which
was one of our caveats, which we'll explain later. But most of our locations had a very low score due to not finding that many macroinvertebrates because of that. So this was pretty much just our results and how we did it. Again, 10 large strides, 85 meters. We repeated this process at 5 locations, but if we could have done more, we would have, because that would have helped our data as well.
Okay. I'm going to talk really quickly about our results. Unfortunately, there's no correlation across each of our compounds that we decided to test. Yeah, I'm not going to read the figure legends, but yeah, there is absolutely no correlation across any of them at all whatsoever. Nothing interesting.
All right. So yeah, in the end, the results that we got for all 3 compounds mostly supported our null hypothesis, which states that there's no correlation between the levels of nitrogen compounds on the macroinvertebrate presence or diversity, and rejected our original hypothesis, which stated that there would be an ideal level of nitrogen compounds, and we would see an increase with that. And then with like lower or higher, we would see lower levels of macroinvertebrate presence. And yeah, our lack of correlation can probably be attributed to the fact that we were taking it from like the same body of water, and we couldn't find that much variation in the levels of nitrogen. And so trying to like establish some sort of correlation based on that proved pretty difficult because we just didn't have that variation. And then an impact of climate change and rising temperatures and sea levels. As you can see in this photo, the lagoon is very close to the ocean, and so while it does happen occasionally that there's like incoming water from the ocean into the lagoon, which is why it is like brackish. It's a mix of salt water and fresh water. Rising sea levels would increase the amount of salt water, and so with like macroinvertebrate presence, like you probably see a drop in population with that. In terms of like its effect on the nitrogen compounds, there wouldn't be that much of an impact there, but just looking at like macroinvertebrate presence, you would definitely see a change. And then comparing like our data to the biological context of the field site, the lagoon is like currently experiencing a lot of algae blooms from cyanobacteria, and that's kind of a process that's kind of increasing right now. And this can be caused by like runoff and like an increase of nitrogen. And because nitrogen is like a limiting factor, right, for plants, and so it'll like often cause like large influx in plant levels, and this can lead to what's called eutrophication, which does often deplete the oxygen levels in the aquatic system, and so there you would see a decrease in macroinvertebrate presence, or that'd be likely. But since we had no correlation there, it's kind of hard to say. It's kind of hard to compare any similarities or differences. But just in general, to help mitigate with eutrophication, a land manager could, coordinate with nearby farms who might be using pesticides or fertilizers, and that is heading into the water that is eventually leading into the lagoon. And so trying to mitigate that. And then for our caveats, yeah, definitely our data could be taken with a bit of a grain of salt. One thing is, as I touched on in our results, like there wasn't that much variation. And so there was no way to reliably change or manipulate our independent variable. We were just kind of hopping around from station to station, hoping that there would be some sort of change, but there was no guarantee to that. And so if we were to like redesign this experiment, having a way that we could like actually manipulate and guarantee a change in those nitrogen levels, maybe we could add it in ourselves to the samples. That could probably help us visualize some sort of trend. But otherwise, we were able to guarantee that variation, and we didn't have that. So, and then another thing is kind of like what the last group touched on. There's human error. Sorting through those the dirt from the DNet was kind of difficult. It would kind of blend in with the brown color, and so I'm sure there are bugs that weren't always accounted for. And then another thing was the usage of the bug chart. You guys saw it was like, I don't know, only like 20 species, so it was very limited. And a lot of times we would find macroinvertebrates in our sample that we just couldn't count as data because they weren't on the chart. And that impacted our data hugely, right? Because we could have like a sample where it was a bunch of bugs, but that would be a bug score of 0 because none of them were on the chart. So yeah, so if we were to do future research, there we go. If we were to do future research, maybe instead of that bug score chart, we could use what's called the Hilsenhoff Biotic Index, which is a similar concept, but is much more comprehensive and includes way more species. And so if we were to use that instead and maybe compare our results and maybe extrapolate some sort of trend from that, that'd be interesting to see.
Okay. Are there any questions? Yeah. Okay. Avery.
Jane was talking about how a more controlled testing environment might lead to like more cohesive results. Was there any outside like research that proved that, or is that just a hypothesis?
It's just a hypothesis.
Sort of similar to this, but like, what was the expected result? I think you may have touched on this, that the nitrogen compounds may have had. Would it have been direct, like impacting bug growth, or would it have been like changing the pH, which increases creating more algae, which changes the pH, which would have then increased?
It would be more direct because nitrogen is like key in like biological process for any living species, like it's in amino acids, right? And so if we had too little nitrogen, right, they wouldn't have those in the formation of amino acids and proteins and whatnot.
But we could look at it as both like direct and indirect, because that comes up in the presentation quiz, where like if there was like too much or too little nitrogen, it would affect the algal blooms, which would completely affect the macroinvertebrate presence and bug score. So yes, you can look at it both ways. Okay. Sorry. Yeah, Mateo.
I'm not sure if you guys talked about it, but like, were the nitrogen like compound levels like pretty normal, or were they to any extreme?
They were pretty normal or very close to 0. We had like all of our nitrogen levels at almost all of our sites were practically like very similar. You can see in our testing, and they were within the normal range. So we were kind of expecting on the lower side of the normal range, but we were expecting there to, as we went through the lagoon, it would change a little bit more, but they ended up not. Yeah.
Do you guys think there would be any compound that would have like a greater effect than other ones, or is it more of like any of them could just have like similar effect or something like that?
Out of the ones we tested?
Out of like nitrate.
Yeah,
or,
like what?
I would assume out of like nitrite and nitrate, just because those directly go into like plant and algal conditions, which in my opinion, that would affect the macroinvertebrate presence and diversity a lot more. So I would assume nitrite or nitrate.
Yeah, we had some sources that.
Because that's where we find the assimilation of the nitrogen cycle.
It would definitely be those, because they have the most direct correlation with the algae, and the algae was one of the biggest factors.
Yeah. Okay. Sorry.
Thank you very much.
Thank you.
Any questions? Thank you guys. Group of 5.
No correlation. It's not funny.
Is that one yours? Yes.
Yeah, I like the name though.
So when you all start introducing yourselves, I'll just have
Guys, Okay, we'll start now. The effect of proximity to Rodeo Lagoon on soil composition by Annie, Rory, Karina, Loire, and Hannah. Our research question is, what is the effect of proximity to the lagoon on soil composition? Our independent variable was proximity to the lagoon, and our dependent variable was soil composition, measured in value and chroma.
our proximity was measured in inches.
Our hypothesis was that the proximity to Rodeo Lagoon affects soil composition. The closer to the lagoon the soil is, the higher its chroma and the lower its value because it has more access to water, which is a solvent for its nutrients. Our null hypothesis is that soil's proximity to Rodeo Lagoon has no effect on its composition. This was our location at Rodeo Lagoon.
Okay, now moving on to materials and methods. So for materials, we used a Munsell color chart, which is a color system that organizes color based on light and darkness on a scale from 0 to 10, and chroma, which is the intensity and purity of color from dull to saturated. These attributes allowed for a systematic and visually uniform way to specify any color serving as a standard for various industries. And then we use a soil core sampler, which is a tool used to collect samples of soil called cores without disturbing the layers of the soil, so we can see it clearly without disrupting that. And then we also use deionized water and a measuring tape. So the methods that we used was, one, we used a measuring tape to find distance from the lagoon. We started a little bit close to the lagoon and then slowly went farther away. And then again, we use the soil core sampler to get the soil cores, as you can see in the image right there, and wet the soil cores with deionized water, compared that to the chart, which you can see in the bottom. And then we did this 10 times close to the lagoon. Each time we went farther and farther away. And then at the end, we repeated this process several times, and we had 30 total samples at the end.
All right, so I'm going to talk about our results a little bit. First, we made two different graphs, one for distance versus the chroma and one for distance versus the value. And we decided to graph the averages because it was a little bit easier to see. It got a little bit confusing with each data point because there was 10 for each, and as you can see farther away at the 100 inches, there was a little bit of a higher chroma, which means that the soil was a little bit healthier farther away. And closer at 25 inches is a little bit lower. The 50, we'll get into this a little bit, but wasn't as consistent with our research. And then on this graph, this is the distance versus value. There's a lower value at the 100 inches farther away, which is the opposite case where lower value actually means it's more healthy farther away. So this will all make sense in a second too. Both of those graphs suggest that distance does have a bit of an effect on form, color, and like nutrients of soil, though it was not as much as we thought it would be, as you can see by the kind of flat slopes. The results did reject our null hypothesis, and distance did, again, have that effect on both chroma and value, which was an indication that proximity was an important factor for soil health. However, the p-value for chroma was 0.52, and the p-value for value was 0.67. And that showed that our data was largely insignificant because, as you can see on the chart, it was not close to 0.05. Okay, now let's talk about what the heck is a Munsell color system. So first, on the y-axis is value, and that measures how light or dark the soil is. And so darker means that it's mostly comprised of organic matter, which is healthier for soil. And so those would be the lower numbers of 2 and then all the way up to 8. In our case, this is the chart type that we use. There's multiple types, all the way up to 8, which would be lighter, and it lacks like essential nutrients and organic matter. And then on the x-axis is chroma, going from right to left, and a low chroma, or chroma measures like the vibrancy of the soil. So you can see in our case, again, we were measuring, it's more like orangey when it has more color. And so a chroma with a low number, such as 2, means that it has like
it's lighter in color. You can see it better at the top. And then on the right is a more vibrant orange color, which indicates that it has more oxygen in it. And so the lower numbers, they're more waterlogged, which we'll talk about more later, but that means that soil's less healthy. So if you recall our initial hypothesis that water would actually help soil health, that turned out to be wrong because water was turning out to have a negative effect on like oxygen levels and nutrients of the soil.
Okay, doke. So, more in the discussion. It makes sense why soil close to the lagoon is more waterlogged. Its chroma would be a lower, less oxidized and waterlogged number. And soil further away from the lagoon is less waterlogged, meaning it can oxidize better, it can drain better, and it's more efficient and better for the organic matter composition in that soil. So it would have the soil further away would have both a higher chroma number and a lower value number. What's specific to our lagoon is that it's a coastal lagoon, and it does have salt in its water composition, and salt doesn't have an effect on both chroma and value for the soil. More salt in soil means that there's more water retention. You can think about when you're sick, sometimes you have to eat like salty crackers to keep the fluids that you have in, and that's the same for salt in the water. And so the brackish water, which is a mixture of salt water and fresh water, is present in the lagoon, could influence the wetness of the soil, which could affect the chroma by making the soil more waterlogged, and the value of the soil by affecting the hue. But because it's a lagoon, it's a mixture, so the brackish water also offers a unique mixture in that it doesn't have the higher salinity level of the ocean, but it doesn't have the lower salinity level of, say, freshwater lakes. So the chroma and value for a lagoon is very similar to its location. Some effects of climate change are rising sea levels, which means that as sea levels rise in the lagoon, that could equate to less drainable oxidized soil because more of the soil would be closer to the water. There's also fluctuations in salinity level. This is a study done about coastal lagoons, actually, I think in Tahiti. It says that it creates sort of a salty soup, which means that because lagoons are low-lying body of water, more of that coastal water would flow into the lagoon, messing up the salinity levels. And also rising sea levels increase the likelihood of flooding and erosion, as well as inconsistent rainfall, such as fog and wind, affects the brackish water composition. You can think on a given year where you get less rainfall, there would be less of a freshwater composition in the lagoon mixture. And also there's an increase recently in more intense storms, which is more likely to cause erosion and pollute the waters, as well as global warming contributes to evaporation rates. The other unfortunate thing is that humans are causing more coastal floods, which again messes up the soil composition as well as the mixture of salinity. So.
Okay, so we did have a lot of caveats in our experiment because when we got to the site, we decided to change our questions. So we didn't have all the necessary tools to conduct our experiment. One of our caveats was that we didn't have full access to the Munsell chart. We had to use a little image of the Munsell chart on a piece of paper, and that image shows how small of a Munsell chart we had. So as you can imagine, like when we got the soil core samples, we couldn't always see the color very well, so that might have impacted our data a bit. Additionally, we measured soil composition very close together. As you heard, we measured at 25 inches, 50 inches, and 100 inches away from the lagoon. And to be honest, our results came in like pretty similarly. And if we repeat this study again, we probably would have taken distances more further away from each other to find more variability in our data. Another thing was that we didn't consistently wet the dry soil samples, and this could have led to the color being inaccurate in some of the soil samples. And another thing we didn't do was we didn't have a plotted out area for each distance we took samples from. So sometimes when we took samples of the soil, there's a lot of plant overgrowth there or rocks around them. So we had to like move a little bit so the distance wasn't always very consistent. To contextualize our research, we came up with a future research follow-up question that connects plant growth and soil. And it's, does soil location and composition affect plant growth? We decided to come up with this question because nutrients in soil has an effect on plant growth, obviously, and we thought it would be interesting to research. Also, we want to note that there's nothing much a land manager could do for this because plants are already used to their environment and it's natural. Okay, so for our ideal results, had we planned for all the caveats, our results would have been more statistically significant. And in our graph, we would have seen steeper slopes, and overall we would have seen a bigger variety and difference in our results. Also, we would have seen that closest to the lagoon would have had a lower chroma, which means the color would have been more dull, and a higher value, so the soil would have been lighter. And conversely, we would have seen that further away from the lagoon, there would have been a lower value, which would have been darker soil, and a higher chroma number for a more saturated color. And just to like re-summarize, we were right that distance had an effect on soil composition, but it's not necessarily because it's closer to the water. Sometimes being closer to the water doesn't necessarily mean that it's healthier, because being too close to the water can lead to the soil being waterlogged, but being too far away from the water can lead to the soil not getting enough water. Okay, here's our sources. Questions? Yeah.
What's like the ideal balance of water retention for soil health, like in between like not having enough water and then being like waterlogged? Is there like a range that's
I don't know if there's exactly a range, but it ties back to like chroma levels if it's too waterlogged. So I think what I would advise future study people to do is test the salinity levels to see like what effect that might have on the chroma data. You know, chroma ranges in certain numbers, and I think that you can figure out what your specific climate is, because it also it varies by location, of course. And so I just think there's a particular balance between what is like a healthy, not waterlogged, oxidized, and drainable soil, and something that's more waterlogged. That
and that would be like a probably 3, 4, or like 3 or 4 is probably a good number.
And was there like a visible change in like the plants that were in the soil that you tested? Like the plants that looked healthier were in the soil that was healthier according to your calculations?
We weren't looking at the plants, we were just looking at the soil, so we weren't paying attention to that, I wouldn't say. Yeah.
That would be something to research further into.
Mateo?
In terms of chroma, like how would the soil of like a lake compare to a lagoon if they are like if they had like the same amount of water retention?
I think, that goes back to the water retention again. So if they have a similar water retention, then it would probably be the same. But more salt means more water, so then it would have,
A lagoon would probably have a lower chroma value than a lake would, because a lagoon has more of a brackish water composition, which means a higher salinity rate than a lake would. So a lagoon would probably have, like we had a chroma value around 2, but a lake might have a chroma value around 5, which means it's more able to oxidize. So.
Ava?
Oh, I was wondering if you guys could clarify like the difference between the two graphs again.
One was just for the chroma, which was that x-axis. So it was the two different, and then this one is the value. And so the chroma is an upward slope because farther away there was or like it's different because one chroma needs to be high and value needs to be low.
Thank you guys very much. Anyone need paper?
Yeah, I got it.
You can use, feel free to go back.
There you go.
Everyone ready? Okay. Stressed out. The effect of solar intensity on plant VPD. So I'm Lucas Detmar.
I'm Mateo Gonzalez.
I'm Elliot Scott.
I'm Luke Waller.
Okay, so for a little introduction, our experimental question was, what is the effect of solar intensity on plant VPD? Our independent variable here is solar intensity measured in lux, which we measured with the digital illuminance meter, which is the image on the bottom. And our dependent variable was the plant VPD in kilopascals or kPa, measured with the VPD gun on the top. Our hypothesis is that we think an increase in solar intensity will result in an increase in VPD. And our null hypothesis is that solar intensity will have no effect on VPD. For some background, solar intensity is simply the measurement of the amount of sunlight going into a certain area, which we measured with the digital luminance meter. VPD stands for the vapor pressure deficit, and is the measurement of it's the difference between ambient vapor pressure and plant vapor pressure. And so the VPD gun calculates everything for us. So we just have to read what it says. But how this is actually calculated is that first, the VPD gun measures the ambient temperature and ambient humidity to find the vapor pressure. And to find the plant vapor pressure, it measures the temperature of the plant leaf. And then we assume that within the plant leaf, it is 100% saturated with water, meaning that the humidity is at a constant 100%. So you only need to measure the plant temperature, and it can calculate the plant vapor pressure on its own, and then it compares the difference between these two values to find the deficit. So for our materials and methods, as I went over, we use the VPD gun and the digital illuminance meter to measure our dependent and independent variables. And then we also use the Picture This and Seek apps to identify our plant species. So for our methods, we would first use the Picture This and Seek apps to identify a plant species. Next, we would measure the solar intensity using the digital luminance meter, and then find plant VPD with the plant VPD gun. Next, we would record all this data down, including all the temperature, humidity, and ambient vapor pressure data, just for consistency. And then we would repeat with another plant species or in a different location. And these are all 7 of our locations that we measured.
Okay, so as we can see with our results across the different locations that we gathered and the 4 different species at varying different light levels, you can see that on average, as solar intensity increases, VPD does as well. And and we know this is statistically significant because after doing our linear regression test, it showed that there was strong evidence with a p-value less than 0.01 and an R-squared value of 0.434. As you can see across the different species, for example, cypress trees, they tend to stay closer to a under 20,000 lux range, whereas other species like narrow-leaf sword fern and the tufted bulrush stay between, I'd say, 10,000 lux and 60,000 lux. And coyote brush are more resilient as they stand between like 40,000 lux and 1,000 lux with higher VPDs on average. And if you ignore the species, you can see it's following the trend line pretty well.
Okay, so now on to our discussion. If we compare our results to to our hypothesis, which was that an increase of solar intensity would result in increased VPD, we would see a positive slope. And as Lucas just described, we saw that across all the plant species, which would mean that our hypothesis is validated. On the other hand, our null would be that there would be absolutely no change in VPD as solar intensity increased. This would call for a flat line, which again, we did not see. So why do we think we saw this? Again, as Elliot stated, VPD is a measure of the difference of vapor pressure inside the plant versus the surroundings. And as we learned about solar intensity and infrared, if there's higher solar intensity, that means that there's going to be lower infrared. And we tried to get a lot of variance in this data in that we started in like a more foggier area, then kind of moved around to a lot of positions in the lagoon to try to get some variance. And again, higher leaf temperature would lead to higher transpiration and vapor pressure, which would lead to higher VPD in cases with more solar intensity. This would mean that the plant is more distressed in that the stomata is actually closed, so it's not really getting the nutrients. And in terms of the biological context of our field site, Rodeo Beach and the surroundings are a pretty like unique area in that there's a lot of microclimates. Like for example, we thought that our the data for our cypress trees didn't vary in terms of solar intensity that much because one, there was a lot of shade, and two, because they tended to thrive in areas that had less solar intensity with more fog and more moisture. And again, all the rest of our species were also, except for coyote brush, the rest of our species were also suited for like these very high coastal humidity like spaces. And yeah. If we were to like do land management of this ecosystem, VPD is pretty key in doing that because understanding how distressed a plant is and like how much moisture it actually retains is very important for like fire mitigation and drought retention. Because if plants are unhealthy, then they're going to dry out, and that's just like very dangerous in terms of fires. It could also be used to figure out the best time for planting and watering, because you don't want to plant some plants in a time where like others are suffering and you don't want to like overwater a plant too. And some factors that may have contributed to different results if we were to do it again for that. If we were to do this experiment in a different place, although we might have similar solar intensity, the fog is very key to like understanding our data, because whereas with high clouds, there's going to be like the same solar intensity, there's going to be different humidity in that actual area. Whereas with low fog, there's going to be more humidity in the air, which will lead to different VPD because it's a measure of the difference in terms of humidity as well. And as stated again, Marin Headlands is very unique, and there's a bunch of streams and stuff that's also providing nutrients and water to these species. Climate change will affect our area in like a number of ways because one, rising temperatures will distress plants. Of course, there's going to be more solar intensity, and that will also mean that the fog's probably going to burn off and the plants are going to be less protected. Additionally, storms and rising sea levels are going to lead to erosion and habitat loss for a lot of these like specially adapted plants and native species. And also storms would kind of do the same thing.
Okay, so now I'm just going to talk about some caveats and things that we would potentially research in the future. Hang on, camera. Okay. First of all, one of the main things that we would like to change in the future is increasing our sample size for all of the species we measured, and also for potentially the species that we had to cut out because there simply just wasn't enough data to actually use it and for it to accurately represent the relationship between solar intensity and VPD. For example, like if you go back and look at this tufted bulrush, you can see how it doesn't really reflect the overall slope and trend of our thing. And I think one of the main, I think this could definitely be mitigated if we had more data, because as we've seen, it definitely fits the biological context for the solar intensity. As the solar intensity increases, for the VPD to also increase. So by having more data, we can more accurately represent the trend line, and we can also just have a more accurate conclusion that reflects the research we've done. Another thing that we would change if we were to do this again is to also ensure that like for each light condition to also have more individuals for that, because
For example, just for the tufted bulrush, we only were able to collect data for 1 or 2 individuals in each light condition. And I think by ensuring that we could have more of those species in those conditions, we could more accurately reflect, like I said earlier. Something also important with our data collection method is the sunshade variation, and specifically how we use the digital luminance meter. One thing that we did with our digital luminance meter is that we decided to use the max and min features and then use that to find an average that we use for our data points. But on like further reflection, we kind of realized that that method was kind of flawed, specifically because sorry, it's distracted me. Specifically because it basically how it works on either the max or min is that it just takes all the inputs that are coming in at a time, and so for example, on the low setting, if it goes down to 21,000, as long as nothing goes lower, it will always stay at that level. So if one of us accidentally blocks sunlight or makes it more available, that could also potentially skew our results, making our data less accurate. So in the future, we could potentially have just use the average setting, like the regular setting, and take measurements every 30 seconds for about 3 potential measurements, and then average that out to get a more accurate representation of the data. And one future question that we thought would be interesting to research would to be actually able to see like how do seasonal differences such as like drought versus heavy rainfall affect the relationship between solar intensity and VPD across the different plant species we measured, specifically because like we are very constrained by time and we are only able to measure in one day. So being able to see how the environment changes over longer periods of time and on different environmental conditions more than just fog would be very interesting, and I think it would also help with our data.
Okay, here's our bibliography, but before we finish, we're going to go through some presentation. We're going to do a presentation quiz. No, let's do a quiz first. Let's do the quiz first. Okay. Who knows what VPD is? Annie?
Vapor pressure deficit.
Yes. Okay. How does high VPD or low VPD indicate a healthy plant? Lucas, would you like to go over that?
I have an explanation for all of you. So VPD is the vapor pressure deficit, and that means as VPD goes up, the deficit increases and it gets more dry. So as high VPD increases, the plant actually becomes less healthy. So low VPD is a healthy plant.
And this is because like when it's at low VPD, the stomata is open so that there's going to be more transpiration and more nutrients actually going into the plant. And why might foggier areas have a lower VPD? Emma.
It's a lot more moisture in the air. It's a lot more moisture in the plant.
Yeah, it's that, and more sunlight's going to get reflected, so it's going to be a little cooler. Okay, now we're doing questions. Rory.
You mentioned that understanding how distressed a plant is good for fire mitigation. Once you figure out what the distress levels of the plant are, what would you suggest the landowner should do?
Well, if there's like obviously if there's unhealthy plants that are like dying, getting all dried out, we would probably recommend just like clearing them, because that can be really dangerous to have all this dry brush around, especially in an area like that, which is hard to access, and that could actually like burn pretty thoroughly. Emma.
Did you see like variability within like plants like trees, like trimming one trend and like brush trimming another trend?
We kind of saw that with the coyote brush, but most of that, how like for example, at the like max lux that we could get, we had our highest data point. But we thought that was mostly because the coyote brush, unlike the cypress, was in areas that were like that had more sunlight. Because for example, the trees thrived in areas with more fogs, and we also kind of measured under the branches. So on average, they kind of had lower solar intensities. But if we were to do this on like a clearer day, I'm not sure what the difference would be. Just based on this, if you look at these two data points, the coyote brush is actually pretty low at a higher solar intensity because it's suited to like a more range, whereas the cypress is suited to a more particular habitat. Okay.
On the list of species, do you know which of those are native versus invasive? And did you notice a trend between those different types of plants? It's okay if you don't know the answer. I wouldn't expect you to. I just didn't know if you did a little bit of further research.
I think, okay, so the cypress was a Monterey cypress, so it's native to California. I'm not sure about, I'd also assume the fern was native, but I'm not sure about the rest. Annie?
You talked a lot about the fog having an effect on the VPD and stuff. So do you think that like if you did it somewhere else that was like not foggy, there would be like your results would be a lot different?
Yeah, I thought they would because even though like fog and clouds both do the same thing, like they both reflect, and you can have the same solar intensity, there's going to be more humidity, and humidity is pretty important for measuring VPD. So with fog, there's going to be more moisture and a lower VPD, whereas with high clouds, it might have the same solar intensity, but there's not going to be that moisture.
Yeah, and I do want to point out that like the different species of plants each can thrive at different VPDs. Like in our graph, you can see that coyote bush can thrive at much higher VPDs, while something like the cypress can only thrive at lower VPDs. And so if you move into an area with less moisture and less humidity, you would find probably different species that are more adapted to live there. And so we believe that you would still find this relationship of higher solar intensity increasing the VPD.
Okay, let's give them a round of applause. So that concludes our presentations this year. Thank you all very much, students, for all your hard work. You asked a lot of great questions while you were building your presentations. MK, did you want to say one more thing?
And we will be sending you a copy of these presentations in an email very shortly. But thank you so much for joining us.